The main objective of this video is to provide you with the theoretical basis of plant tissue culture, leading to demonstration of practical aspects where applicable. The whole video will take about 60 minutes, but is divided into several segments, so you can watch the segments as you progress through your tissue culture activities. To start with, let us look at the definition of plant tissue culture, or as is also known, the in vitro culture. It is the sterile culture of plant cells, tissues, or organs under conditions which lead to cell multiplication or regeneration of organs or whole plants. The important points to pick from this slide is that all microorganisms, such as fungi, bacteria, and viruses, as well as other pests, insects, nematodes, are excluded. That optimum environmental conditions with regard to physical as well as nutritional and hormonal factors is provided, and that the whole process occurs on a micro scale and in vitro. Of course, in vitro, which literally means in glass, came into being because initially, at least, glass vessels were used for the culture. There are several benefits attached to plant tissue culture, and these are summarized in the next slide. To start with, the number of genetically identical plants recovered from a single stock plant is greatly increased. Virus index plants may be obtained through tissue culture, for example by meristem culture. Stock plants may be maintained in vitro where glasshouse space for maintenance of plants is at a premium. This is referred to as germplasm maintenance. Tissue culture techniques may be used to obtain hybrids from incompatible species through embryo or ovule culture. Haploid plants may be obtained through anther culture. We can produce plants all year round using tissue culture techniques. Vegetative propagation of difficult to propagate species may be achieved by tissue culture. Production of new variants may be also achieved using mutagenic compounds or radiation. And finally, it is an important tool of research in many areas of plant sciences. The idea of plant tissue culture goes back to the turn of the century when a German scientist put forward the theory of cell totipotency. Totipotency is the potential or inherent capacity of a plant cell or tissue to develop into an entire plant if suitably stimulated. Totipotency implies that all the information necessary for growth and reproduction of the organism is contained in the cell. Although theoretically all plant cells are totipotent, the meristematic cells are best able to express it. This means that every part of the plant has the ability to produce a complete plant and thus be used in the process of tissue culture. The next two slides demonstrate the parts of the plant that we can use as starting plant material, and as we can see from these slides, basically any part of the plant from roots to stem to leaf, buds, floral parts, seeds and embryos can be utilized as starting material. There are many types of in vitro culture. These are briefly summarized in the next slide. Culture of intact plants, for example, seed culture in orchids. Embryo culture, for example, mature embryo culture. Organ culture, for example, meristem culture, shoot tip culture, root culture, anther culture. Callus culture, single cell culture, and finally, protoplast culture. The area of micropropagation is, however, what we endeavor to cover in this video. Micropropagation is the true-to-type propagation of a selected genotype using in vitro techniques. Several stages are involved in micropropagation, each of which are influenced by an array of physical, nutritional, and hormonal factors. Micropropagation only makes sense when adequate starting material is used. Therefore, the choice of stock plant material cannot be done indiscriminately. For most ornamental and for fruit production, the starting material is an elite plant selected for some phenotypic characteristics. For example, the flavor of the fruit, the color of the flower, the shape of the bush, etc. In forestry and in vegetable production, the elite material will either be a selected phenotype, that means a specific plant, or this plant will be the bearer of elite seed, which will be used as starting material for a more or less extensive micropropagation program. The stages of micropropagation is summarized in the next slide. Stage 0, the preparatory stage. Stage 1, the initiation stage. Stage 2, the multiplication stage. Stage 3, the rooting stage. And finally, stage 4, the acclimatization stage. And we endeavor to take you through these stages by use of demonstration. Stage 0, or the preparatory stage, is about preparing the stock plant for micropropagation. Two principal considerations are involved in handling stock plants. One is reducing the potential for contaminant by fungi, bacteria, viruses, and other pathogens. The other is control of the physiological conditions of the starting plant material. This starting plant material is also called an explant. In general, stock plants should be healthy and actively growing, not in or entering dormancy. Pruning or use of supplementary light for increasing day length is sometimes useful in maintaining active growth. Ideally, stock plants should be grown in a glasshouse. Field-grown materials are often very difficult to disinfest and establish clean cultures in vitro. Not only growing plants in a glasshouse will reduce the population of microorganisms which are living on the surface of the plant, it also helps to produce quality plants for stage 1 due to regular fertilization, use of fungicide and insecticide. So, at the preparatory stage, we need to ensure that the stock plant is growing vigorously and is free from pests and diseases, especially the microscopic pests such as mites and thrips. By establishing the stock plants in a glasshouse, we can ensure that the plants have been fertilized properly, have been sprayed with pesticides and fungicides, and have been pruned so that new, young, vigorous growing shoots are established. Another important point is that under glasshouse conditions, we can control the humidity in the glasshouse. Generally speaking, a dry glasshouse will harbor less microorganisms. The hygiene in the glasshouse can also help to keep plants clean. As a general rule, plants destined for micropropagation should not be watered overhead. All the points we have just discussed will aid in reducing the infestation at the next stage, which is the initiation stage. So to summarize to here, the objective of this stage of micropropagation is to produce healthy, vigorously growing plants free from pests and diseases. Now let us look at stage 1, which is the initiation stage. The objective of this stage is to successfully introduce the desired part of the plant into the appropriate medium in the test tube or any other culture vessel that we use and produce clean cultures. A culture medium consists of all the mineral and organic nutrients that the plant requires for sustained growth. These are all the macro and micro elements, the vitamins and other growth factors such as myoinositol, glycine, nicotinic acid, pyridoxine, thiamine, etc. To this mixture, we also add sugar as source of energy and plant hormones when necessary, as well as a gelling agent. To prepare a batch of media, we can either use pre-mixed media such as Morishige Scooch, Gambur, White, etc., which can be purchased from chemical companies such as Sigma, which has a wide range of media in powdered form. The only thing we need to add to this mixture will be sugar, plant hormones, and the gelling agents where necessary. If, however, we are doing a lot of media preparation with various combinations and permutations of minerals and organics, we may need to opt for preparing our own stock solutions and use them as required. Whilst this provides a lot of flexibility, it may work out to be a bit impractical for small labs. One needs to have all the chemicals and a sensitive balance to measure accurately quantities required, a fridge to store the stock solutions, and suitable containers to store these stock solutions in. The final choice will very much depend on the quantities of the media you are preparing and the facilities in which you are working. To prepare media, simply measure the quantity of stock solutions or add the correct amount of powdered medium to deionized water. It is important that the water used for tissue culture be deionized. Deionized water can be purchased from many supermarkets or water purification companies. Large laboratories normally have their own purification plant. To this mixture, sugar is added and the pH is checked. As a general rule, the pH of tissue culture media is adjusted using either a weak acid solution like 1 normal hydrochloric acid or a base like 1 normal sodium hydroxide. A pH meter is necessary in big laboratories, but if you are a hobbyist and have no access to a pH meter, pH paper with narrow range would be just fine. As a general rule, the pH of most tissue culture media is set at around 5.6 to 5.9. The gelling agent is then added. There are various gelling agents that can be used. For example, agar, which is extracted from the cell walls of a number of general red algae, which produce an opaque gel and, generally speaking, is dissolved on a hot plate with magnetic stirrer for continuous stirring of the agar. Gelrite is from bacterium Pseudomonas elodea, and gel cream from the walls of a cultivated red algae. Gelrite and gel cream produce a clear gel and can easily be cold dispensed as long as the mixture is kept in suspension by constant stirring. Otherwise, the gelling agent will settle to the bottom of the beaker, resulting in a batch of medium which is not homogeneous. This means that some containers will have a lot of gelling agents and some will have very little in them. Once the gelling agent is added and dispersed or dissolved thoroughly, the medium can then be dispensed into culture vessels. There are a wide range of culture vessels: the glass jars, the plastic ones, which are autoclavable, the plastic ones, which are not autoclavable, and can be purchased in sterile packs. These have been sterilized using radiation. For these ones, media is bulk autoclaved, and then the correct quantity is dispensed into them under sterile conditions. For others, the culture vessels are capped and autoclaved for 20 minutes at 121 degrees centigrade at a pressure of 103 kilopascal. This pressure and heat will destroy all microorganisms which are in the medium and the culture vessels. A household pressure cooker may be used instead of an autoclave in small hobbyist operations. Another method of sterilization of media is filter sterilization. In this case, a membrane which is made of cellulose acetate or cellulose nitrate with 0.22 micron pore is used. This is used only when we are dealing with compounds which are heat labile, such as some of the plant hormones used in tissue culture, for example, zeatin, gibberellic acid. All the instruments and equipment needing to be used must be either autoclaved or soaked in 95% alcohol. For example, the hypodermic syringe, the container in which the compound in question is going to be filtered into. The compound in question is drawn into a hypodermic syringe and then are passed through the filter like this in a laminar flow cabinet or any other clean environment. The correct quantity of the hormones is then added to the medium after it has been through the autoclave and cooled to approximately 50 degrees centigrade. In this temperature, the agar and other gelling agents still are molten. The medium is then shaken vigorously to mix the added compound before being dispensed in the appropriate culture vessels. Media can be prepared ahead of the time and be stored in a cool, clean cupboard in dark or inside a refrigerator. The next step in stage 1 of micropropagation is preparation of plant material. The plant material is harvested ideally first thing in the morning when the plant cells are all turgid. Shoot segments, leaves, petiole, bulb scales, etc. can all be used as indicated before. The plant material then is washed under running tap water for 20 minutes to 2 hours, depending on the source of plant material, that is, whether the plant material is field-grown or glasshouse-grown. If the pieces of plant material used are small, wrapping them in a piece of gauze or chuck's cloth will hold the material intact during the sterilization. Then, the plant material is rinsed in 70% alcohol for 10 to 30 seconds. Alcohol not only kills many microorganisms residing on the surface of the leaves or buds, it also acts as surfactant. Third step is to wash plant material in a chlorine solution, such as sodium or calcium hypochlorite. These are sold as commercial household bleach, such as White King. This is a strong chlorine solution and requires to be diluted. We need to dilute this solution to 1/4 strength. This gives us a chlorine strength of 1% weight per volume. It is best to prepare this solution fresh. To this solution also add a drop of wetting agent, such as Twin 20 or Triton or any non-ionic detergent. The surfactants or the wetting agent will improve the surface coverage. The plant material can be washed in this solution for 5 to 20 minutes and sometimes even longer. Modification to this basic procedure that can be used to improve efficiency include mechanical agitation and use of vacuum as demonstrated here. Finally, everything is taken into the laminar flow cabinet and the hypochlorite solution is discarded and the plant material is rinsed 2 to 3 times in sterile distilled water. The laminar flow cabinet or transfer hood is a cabinet in which air sucked from outside has passed through very fine filters called HEPA. that stands for high efficiency particulated air. This filtering system ensures that the airflow over the working bench is completely sterile. In fact, HEPA filter catches any particle which is bigger than 0.3 micron in diameter. Since there is a continuous airflow through the inoculation cabinet, it is particularly impossible that anything can pass from the outside room into the cabinet itself. When the cabinet is not in use, the cabinet can be closed from the outside air by use of plastic shields like this. The laminar flow cabinets are also fitted with a UV light, which can be used prior to the time one needs to use the cabinet. UV light is a germicide. It also causes skin cancer and affects the retina in our eyes, so it must only be switched on when the shield is in place. The UV light must be turned off prior to the removal of the shield. The motor, which draws the air, must begin to operate. Now, if you do not have a laminar flow cabinet, a fish tank on its side, like this one, which has been swabbed and sprayed with alcohol, would do just fine. But you have to bear in mind that for a commercial lab, these will not be sufficient. The operator should also be cleaned up, hair tied back, jewelry removed, hands washed and surface sterilized using either 70% alcohol or other compounds such as hexafoam. A clean lab coat and a face mask will reduce chances of accidental contamination. The plant material, as a general rule, is cut to the desired size on a moist, sterile filter paper. It is important to note that any item used inside the laminar flow cabinet must be sterile. That is, the dissecting instruments such as the scalpels, the scissors, the forceps, and indeed the petri dishes or the papers you use to cut the plant on. Now let us look at the preparation of the explant. The exact method for preparing the explant varies with the kind of explant. In general, all procedures described up to here should be followed. If shoot tips are used, remove the disinfested shoot pieces from the rinsing solution and cut away the enclosing leaves with sharp scalpels or needles to expose the immature shoot tip. Removal of only meristem dome and a few leaf primordia requires very fine micro tools or scalpels and use of a dissecting microscope. If the entire shoot tip is used as explant, the operation is less exacting. In either case, the work must be done quickly and the pieces must be inserted on the gelled medium quickly to minimize desiccation. In other cases, leaf segments, petiole segments, or root segments may be cut following the same instructions. All dissecting instruments need to be sterilized in between cuts. To do that, we can flame them in 70% alcohol and then allow them to cool. Or we can use a bead sterilizer like this one here. The bead sterilizer heats a series of glass beads to 250 degrees centigrade. We only need to insert the instruments in them for about 5 to 10 seconds to kill any microorganisms which may exist under. There are other equipment used for this purpose, for example, bacticinerator. No matter what it is called or what you use, the objective of all these are to kill any microorganisms which may have survived the process of surface sterilization of the plant material. At initiation stage, it is desirable to use small vessels and put only one explant into each vessel. The reason is that if you use larger vessels, which you can put more than one explant in, if one of these explants become infested in the vessel, you must discard the whole lot, whereas in a small vessel, you contain the infestation of the one explant to that explant only. These cultures are then transferred into the growth room with diffused light. The light intensity in a growth room like this is about 50 micromole per square meter per second, which is about 1/50th of sunlight in a cloud-free day. So it is not very strong. The photoperiod, as a general rule, is set at around 16 hours day and 8 hours night, although this might be altered depending on the species and the time of the year you are doing the culture. The temperature of a growth room for most temperate plant is 25 degrees centigrade. 28 degrees for tropical plants like banana, and 18 degrees for alpine plants. I need to indicate here that in most micropropagation operations, the explants are placed initially on a medium containing only basic salts and sugar, but without hormones. If the explant contains phenolic or other substances that exude from the cut surface and inhibit development, charcoal, ascorbic acid, citric acid, or other substances may be added to the medium to prevent the effects. Using antioxidants such as ascorbic acid or citric acid in the preliminary wash also may be useful. Use of liquid medium in the initial stage and frequent changes for several days may help leach out the phenolic compounds. The contaminants consisting of yeast and various species of fungi and bacteria usually appear on the medium surface within a few days or a week as white or opaque slime or as variously colored colonies. Sometimes black spores and mycelium can be observed. When these contaminants can be identified visually, cultures containing them are quickly discarded. Contaminated culture vessels should be sterilized in the autoclave without removing the cover to avoid contaminating the laboratory. Some bacterial contaminants, such as Bacillus, Erwinia, or Pseudomonas, sometimes remain inside the plantlets without being detected until late in the culture period. These are the cause of what we call the latent infestation. Procedure to identify this contaminant may need to be included in a large commercial lab. Antibiotics are sometimes used in the medium to improve control of bacterial growth. However, the prolonged use of antibiotics should be avoided. Plant preservative mixture, or PPM as is also known, is a new product which reduces infestation rate dramatically when added to culture medium. Clean cultures from this stage are then transferred onto the next stage, which is the multiplication stage. This stage usually takes about 4 weeks. To summarize, the purpose of stage 1 is to produce axenic or clean cultures. Now let us look at stage 2, which is the multiplication stage. The function of this stage is to induce multiple shoot development. This may involve the stimulation of axillary shoots, the initiation of adventitious shoots on excised shoots, leaves, bulb scales, flower scapes, cotyledons, and so on. The initiation of callus from the cut surface. Therefore, the medium selected varies with species, cultivar, and the type of explant to be used. Control of development is achieved by manipulating the levels of auxins and cytokinins in the medium. If axillary shoots are desired, a moderate levels of cytokinin, such as BAP, kinetin, or 2IB, at concentrations of 1 to 5 micromolar are used. The auxin level must be kept very low or omitted altogether. To develop adventitious shoots on the explant, a somewhat higher cytokinin level is needed. For callus formation, increased auxin levels are also needed, but must be adjusted to an appropriate level of cytokinin. The culturing is done by cutting off the shoots and transferring the base to the new medium. Elongated shoots may be laid horizontally on medium surface, acting as a type of layering, often stimulates lateral shoots to develop. The number of propagules produced for transfer every 4 to 6 weeks varies from 5 to 25 or more from each explant. Multiplication stages may be repeated several times to build up the supply of material to a predetermined level for subsequent rooting and acclimatization stages. During these multiplication stages, off-type propagules, like these ones here, sometimes appear, depending on the kind of plant and method of regeneration. Transferring cultures onto a hormone-free medium sometimes overcome this problem. So, to summarize to here, the purpose of multiplication stage is to produce numerous shoots for transfer to the next stage, which is the rooting stage. The purpose of this stage is to prepare the plantlets for transplanting from the artificial heterotrophic environment of the test tube to an autotrophic free-living existence in the greenhouse and onto their ultimate location. This preparation may involve a change in the physiology of the plantlet so as to stimulate photosynthesis, nutrient and water absorption through roots, and resistance to desiccation and to pathogen. At this stage, propagules can be handled in 3 basic ways. First, individual cuttings can be made and planted directly into a rooting medium in mist or under high humidity with or without rooting hormones. In other words, we treat them like soft cuttings. Second, individual propagules may be recultured into new containers in a sterile medium with reduced or omitted cytokinin, an increased concentration of auxin, and often a reduced organic salt content. For some kinds of plants, adventitious roots then develop readily on these propagules. Better root hair development may occur in an aerated liquid medium, for example on sorber rods or filter paper bridges, than in an agar medium. With other plants, rooting is best if the propagule is kept in the auxin medium for only 1 or 2 days and then transferred to an auxin-free medium, or the propagule may simply be dipped into a rooting solution immediately and inserted directly into an auxin-free medium. The third method is to include an elongation phase here by placing the propagules into a medium without cytokinin or at very low levels of cytokinin for 2 to 3 weeks, and in some cases adding gibberellic acid. This may be necessary when numerous short shoots are resulted from the multiplication stage. Selection for uniform propagules and discarding the inferior and abnormal or diseased plantlets should be made at the start of this stage. Sometimes cultures deteriorate with time, lose leaves, fail to grow, develop tip wear, go dormant, or lose potentiality to regenerate. Plants of a species having an inherent dormancy or rest requirement may need to be chilled to stimulate new growth and elongation. Other problems indicated may require detailed experimentation to ascertain the reasons for lack of growth in cultures. So, to summarize, the objectives of this stage are to select vigorous and uniform plantlets and induce root formation either in vitro or in vivo. This is the final and most critical stage in micropropagation. Plants grown under in vitro conditions have an anatomy and physiology which makes them unsuitable for survival in the outside environment. These plants have been growing under high humidity, low light intensity, constant temperature, aseptic conditions and readily available nutrients, resulting in leaves which do not have epicuticular wax. The stomata are either non-functional or not as receptive to changes of humidity as are the in vitro grown plants. The vascular connections of stem and leaves are not extensive, and more importantly, these plants are not autotrophic. That is, they cannot synthesize their own food at this stage because their photosynthetic machinery is either not activated or partially activated. Indeed, in vitro grown plantlets are called heterotrophic or mixotrophic. Because of these reasons, the in vitro grown plantlets need to be introduced to the outside environment in a gradual manner. As a general rule, the plantlets are removed from the culture vessel in a room away from the direct sunlight. The gelling agent is washed away completely to remove a potential source of contamination. The long roots and shoots are slightly trimmed, and the plants are then transferred into a suitable pasteurized potting mix. The potting mix may be a mixture of sand, peat, and vermiculite. No fertilizer is added to the pots at this stage. At all times, desiccation must be avoided, and that can be achieved by frequent water spray on the plants. Initially, plantlets should be protected from desiccation by placing them in a glass house with reduced light and high humidity. This may be achieved by use of foggers or misters, or use of wet tents. To reduce light, a shade cloth can be used. Shade cloth come in various densities. Up to 70% of the light may need to be eliminated in the first few days. Gradually, the light intensity is increased and the humidity is decreased until such time the plants have developed functional roots. The temperature in the glasshouse should be maintained as close to that as the growth room, where the plantlets come from. Nutrient of the potting mix is another limiting factor. To play it safe, do not provide any nutrient in the potting mix until plantlets have been removed from under the mister. This could be about 2 to 3 weeks, after which time a slow-release fertilizer, such as Osmocote, can be added to the pots. Dilute liquid fertilizers, such as Aquazole, could also be included on a weekly or fortnightly basis. The length of the time taken to acclimatize plants varies depending on species, but generally it takes between 4 to 6 weeks. Although at this stage the plants are grown under non-sterile conditions, the hygiene is still very important. Diseased plants must be eliminated quickly and the area be washed with a suitable disinfectant. The acclimatized plants are later transferred to larger pots or to field as required. So to summarize, the objective of this stage is the production of hardened-off plants that can cope with the outside environment. Producing plants through tissue culture is now an important method of asexual reproduction in many horticultural plants and an adjunct in plant production through genetic engineering, which will dominate plant production scenes in years to come. We hope that this video has provided you with some fundamentals on this expanding area of plant production.